Anthony Kansas. He started here in summer 22. He started originally in Mary Beth Lappis's lab, who was online. That's why I gestured this way. He was instrumental in setting up the 480 plots, or the deciphering diversity experiment. Like, we could not have done it without Brandon at all, I think. So, however, this presentation is on his own research that he developed kind of around that project. It's not really that project. It contributes to, but your own unique ideas. So that is especially nice. And I probably don't need to tell you, but just in case, we'll hold all comments and questions till the very end. I don't even have an embarrassing story to start with, but all right, take it away.
All right, thanks for that introduction, Laura. Welcome to my event. It's titled Testing the Effect of Source and Environment on Native Seed Germination with Seed-Based Restoration Grasslands.
Oh yeah, click back on the slide.
And first off, I would just like to take a moment and acknowledge the people that made this research and my experience at South Dakota State University possible, including members of my committee, members of the NRCS grant this research is funded by, the entire Perkins lab, and those who graciously decided to help with this project, and also the ever approachable faculty at the NR. I would also like to recognize that this research was done at the Perkins campus of SDSU, which is part of the traditional, contemporary, and land of the Rochetti Shakowin people. And I hope that this research can, in some way, be implemented to benefit those indigenous communities. Apart from that, grasslands, as we know them, are these wonderful rolling hills of vegetation that we know and love in South Dakota. They host all sorts of different plants, wildlife, amphibians, birds, all the way down to the smallest microbes and macroinvertebrates. And welcome. One more seat in the presentation. And these plants and wildlife have obviously lived and evolved in this ecosystem, relying on its resources for food and habitat for millions of years. But if we lose these grasslands, it's not too far off that these species will also disappear with those grasslands. And so because losses of the grasslands, the Great Plains are an endangered ecosystem, and they have been greatly suffered great losses. And within the Great Plains, there's also the Northern Great Plains, which contains some last tracts of remnant prairies. This map to the right depicts the historical range of North American tallgrass prairie, and the dark green spots represent places that still exist or are intact. Some of the largest intact pieces of these prairies are in foothills of Kansas, and also in the Prairie Coteau region in northeastern South Dakota. These grasslands are habitat for many endangered species. A couple to be highlighted, like the rusty patch bumblebee and Dakota skipper, federally endangered. And losses of these native plants and the disturbances that the communities evolved with have often been, or mostly anthropogenic in cause. This can be in the form of converted to row crop agriculture, extensive overgrazing, or changes in the climate, or the introductions of invasive species that have native. Hopefully, or luckily, lots of these native communities at least be somewhat mitigated by restoration. restoration is something that looks similar to having a monoculture of an invasive species that we try to remove and replace with native plants in the hopes that they also attract other native plants and even wildlife. So apart from just the beauty of restoration and restoring natural habitat, it also provides essential ecosystem services and benefits, such as seeing soil health, when we think of like carbon sequestration and capture, and water filtration and infiltration, habitat and food for wildlife and pollinators, and productive forage for grazers, and a little bit of recreation if you are a birder or hunter, things like that. Restorations are also good at discouraging the spread of invasives by taking up the space that invasives might otherwise invade when native seed is used for restoration, which we've seen in the past when we tried to use invasives or non-native species to address some of the concerns that we are, some of what we're trying to do with native restoration, things like wildlife habitat, soil erosion, aesthetics. Current priorities and initiatives within restoration include looking into the management and planning of that restoration. Important topics right now are looking into climate adaptive restorations. So how can we prepare our restorations to not just work in the current climate, but also maybe a future climate. Consider the site limitations of things like the soil type, the topography, or even the habitat connectivity of our restorations, while also looking to changes in disturbance and weed pressures. Some priorities of research are systems are in dry and subtropical grasslands, and also looking into effects of post-restoration management. But namely, what this talk is going to be looking into is the importance of seed and the limitations that we have for seed and restoration. So in order to have successful restorations, they need to be self-sustainable, meaning they need to have some type of recruitment of those native plants, and you can't get to the recruitment if you don't get past the first stage, which is germination of those seeds on the landscape. And the National Academies did a big giant report on native seed and asked a bunch of survey questions and answered a lot of just things about native seed. And when they asked state departments what they're using native seed for, almost 90% of them are saying that it's going towards creation or restoration of wildlife habitat. And then further down on some of those percentages, there are other types of restoration and landscaping, but mainly wildlife habitat. And then when asked where they get those seeds from, majority of them, 96%, are using commercially bought seed from seed providers, nurseries, things of that matter. And seed-based restoration is a popular technique used because it has high feasibility in that you can directly seed plants in the landscape with direct broadcasting or seed drilling. A lot of places have pre-made conservation mixes that you can use and put in your cart and go with it. And they also have a lower cost if you applying them over a large geographic area compared to other methods. Some of the challenges that we have faced with seed-based restoration, though, is that often low establishment to the scene, and finding and sourcing these appropriate seeds is a big challenge. And poorly understood germination also contributes to that low establishment of not maybe knowing exactly what all of our seeds are going to do. When we look at the availability of seed across the northern Midwest, the upper Midwest, we can see that it's not evenly distributed. We have higher concentrations of available seed in states like Iowa and Minnesota, but the states to the west are much more sparse. They don't have as many available sources to use. And this was reflected when, in that same National Academies report, when they asked what biggest barriers to doing seed-based restoration were, was that availability of seed in the time frame that they needed is often the biggest hurdle. So people, or not people, but restoration practitioners have been looking into trying to get locally adapted seed for the best success of their restoration. And one of the resources that people have been using are these provisional seed transfer zones, map built together by a study with U.S. Forest Service. And they try to answer the question of how far and to what extent can seed safely be transferred and not suffer from maladaptation to different environments. They answer this by dividing out the U.S. into various bands of minimum temperature, humidity, and level 3 EPA ecoregions, which account for the kind of topography and geography or geology of those regions. And some of the methods you can go about when trying to source your seeds, where you get your seeds from, is you can go down the route of focusing on local adaptation, buying the most local seed to your restoration, or you can prioritize genetic variability. And there's overlap between the two, but they both have their pros and cons in that locally adapted restorations have a decreased chance of outbreeding depression and are highly adapted to local conditions, but they might suffer from that lack of genetic diversity and might be prone to inbreeding depression. Whereas when you prioritize genetic variability, the seed might be less locally adapted, but has a lower chance of that inbreeding depression and a higher chance to adapt to a changing climate in multiple directions. And that can kind of be visualized with these diagrams, with the dark red representing more locally adapted and lighter reds arrows being less locally adapted. The further you get to this goes for a restoration site. So seed germination is defined as the emergence of radicle or some type of endosperm material from the seed coat. And unfortunately, it's neglected in a lot of studies in community ecology. It has a longer history of research in agricultural studies because we obviously use these things for crops. But regardless, germination is crucial to establish our restorations. And that's because the seed is most vulnerable at that life stage of being a germinate, not a seed, not a seedling, but between there. And the timing of when the seed germinates matters a lot in its how it might go forth with growth and reproduction and the timing of that. And there's support that locally adapted seed performs better because it reduces these risks of phenological changes. A big importance point with phenological changes and when they occur is if you have seed that germinates at the wrong time when adverse weather conditions are prone to happen, there's a good chance that the seed is less likely to succeed. And another pattern we see is that germination rate is not consistent across latitude. We have a couple papers that exhibit that. My objectives of this study are to compare final germination percentage and germination rate of seed from 13 perennial prairie plant species, and secondly, determine if germination metrics of these species express evidence of local adaptation to environmental conditions. And those environmental conditions we're putting them to is germination temperature, incubation temperature. My hypothesis that seeds will exhibit differences in germination metrics depending on source and environment to which they're exposed. And secondly, the seed germination rate will increase with proximity of that seed source to polar latitudes. This is a table of the species that I used. I selected these species based on commercial availability of the amount of seed, but also how, I guess, multiple sources were available. So there's a lot of cases where you could look for seed and they would all come from very similar locations, even though they're from different seed providers. But these species span across 7 different plant families with 1 to 3 species in representing each family. This is a map of where I got my seeds from and the original seed transfer zones that they originated from. You can see that there's a pretty decent spread from states North Dakota, Minnesota, all the way down to Nebraska and Colorado, one source there. The northern species are triangles, locals are circles, and diamonds are the southern species. And we took all those sources, or I took all those sources, the seed, and put them into different temperature treatments that were based off these 3 locations, one in Brookings, south of Brookings, and a really pretty far northern location. So 3 temperature treatments, we had a cold, intermediate, and a warm. And I pulled the climate data of those locations off of like minimum and maximum temperatures based on the 30-year normals for the month of May. And so an example of what this looks like would be Sorghastrum nutans, Indian grass, has these 3 different sources, and we put each of those 3 sources to the north into all 3 different temperature treatments. Same with the local and the south. And when we factor all this out, we have 9 species or 9 source treatment combinations. And setting those up in the fridge or in the incubators, they have 3 sources within each fridge for each species, 5 reps, so 5 bags of seeds, and within each bag, 25 seeds, but 5 reps per source treatment combination. So I started tracking germination, recording new germinates every 2 days. So 3 times for 6 weeks, 42 days total. Moldy seeds were removed from the analysis, and any species that at the end of the experiments that had 0% germination were removed for obvious reasons, which brings our total species included in the study down to 11. Stats. So all statistical analyses were done in R and R Studio using various packages, with one of them being germination metrics, which I used to calculate final germination percentage and just germination rate. Final germination percentage is the total accumulated number of seeds that germinated divided by the total number of seeds put in that bag. Germination rate is a measure of seed germinated per unit of time, so a speed of germination, if you will. Sometimes germination rate gets used interchangeably with final germination percentage, but for the purpose of this study, final germination will be the percentage and germination rate is rate, speed. So I did an ANOVA of all those different sources and temperature treatments in each species with source and temperature treatment as the independent variables and germination rate, final germination as the response variables. For the species and response variables that did not meet assumptions of ANOVA, I did an assigned rank transformation ANOVA, still look at if there are differences. And for species that did not experience interaction effects, I looked at main effects using Tukey's honest significant differences and assigned rank transformation contrasts. And then lastly, to look at latitude and germination rate, I used a Spearman rate correlation test to see how well these 2 variables of latitude and germination rate correlated. So how might we expect these different effects to be noticed? If we had no effect and we're measuring germination, what we'd expect to see would be all three species from three sources responding similarly within source, but also across the temperature treatment. It'll look like a flat line of some degree. We're looking at if we saw differences in main effect of source, that would look something like those three lines of sources, which separate from each other and differentiate, with ones being higher than others. For temperature treatment, we would expect to see shifts along the treatments, but that sources would still be responding in similar, like parallel fashion. And for both source and temperature, we would see the sources separate each other from each other, but also respond similarly across the treatment.
Brandon, how many replicates did you have per source per treatment? Thank you. I missed that.
That's
Thank
okay.
you.
And so the interesting, or the part that I'm most interested in is this interaction effect. So what those would look like is if species or the sources respond to sources respond to temperature treatment differently for each temperature treatment. And this can look like the sources and the lines that we draw between them having different slopes that if you extrapolated them might cross at one point, but also points of crossing within the graph are big indicators of interaction of those effects. So results of what we found, or what I found was there's significant interaction between temperature and source. Final germination, there's 7 of the species included out of the 11. Out of those species that did not have interaction effects, saw main effects of source for the remaining 4. And you can kind of see that visualized by them separating, those lines separating themselves, and then the interactions having crossing slopes like that. And then 2 species had significant main effects, temperature. Looking at germination rate, we see their interaction between temperature and source for 4 of those species, with 2 of them being grasses, 2 of them being forbs. And for the species that did not have main significant interaction effects, we saw main effects of source 5 species, these 5 species, and main effects of temperature, these 4. So there were species that had main effects germination rate than final germination percentage. And then final germination percentage had more species with significant interactions. Digging into the main effects a little bit deeper with these box plots, the final germination by temperature treatment, you can see that for the 2 main effects that we can look at, there is higher final germination percentage in the warm temperature treatment and lower in the cold, with intermediate having qualities of both. For final germination by source, we see that 3 out of the 4 species with Dalea villosa, autumnale, and Sorghastrum nutans have higher final germination percentage in the north compared to the south sources, but it's not seen in Symbiotrichum ericoides. So no like clear consistent patterns of how source affects, but leans somewhat towards north sources having higher final germination percentage. Germination rate by temperature treatment. There's a 3 out of the 4 species at higher germination rate in warm temperature treatments compared to the cold, with Dalea villosa being similar across temperature treatment. And for source, we see that 3 out of the 5 species had higher germination rate in the north source compared to the south. And one of the two had higher germination in the south, which is Helenium grandiflorus. We'll touch back up on that later. But again, no clear, consistent results of how source responds to germination rate responds to source. And lastly, when we look at the correlation of latitude and germination rate, we see that there's a slight positive correlation germination rate and latitude. And I included the hypothesis of being polar region because we only looked at species within the northern hemisphere, but this pattern might also be in the southern hemisphere. So as you get further from the equator, increase in latitude. Lastly, to kind of summarize both germination rate and final germination, there's an interaction for both responses for 3 species: Udula pendulus, Cytoxbrama, Helenium villosus, Hairy Wildbright, and Leatris legicilus, Rocky Mountain, Blazing Star. There was no interaction for germination rate or final germination percentage for Dalea villosa, Helenium autumnale, and Sorghastrum nutans. So overall, 8 out of the 11 species demonstrated some type of interaction between source and the environment that they were exposed to. This suggests that most of these species have local adaptation that affects their germination. However, there was no clear patterns of how source responded across species. There was trends, but no significance. This may indicate that there's a need for, when you're sourcing seeds for restoration, use multiple sources, because if you're purchasing a local ecotype and expecting it to perform the best, there's a chance that it might not be performing the best, and it's better to have a safety net of multiple populations. And this could be partially explained that seed raised by commercial producers has been shown to adapt to farming conditions after 8 generations. And I assume most seed providers are following that rule, but I don't know that there's a rule that says they have to end up every 8 generations, so we'll see. Seeds germinated at a high rate in warmer conditions. This could potentially be explained by seeds having a higher mitotic rate of cell division in warmer temperatures up until a point where it's too hot, cells obviously lose that rate and it drops off. But there's a pattern we saw and other studies have seen in their germination trials. There was a higher germination at polar latitudes. This might suggest that these polar latitudes, because they have shorter growing seasons, amount of time for the seed to germinate, grow, and reproduce, that window is smaller. But it could be a strategy that restoration practitioners could implement. Say you get a late start on your seeding, and if you're wanting to implement a northern source anyways, that one might respond to those warmer temperatures quickly, and you could at least get plants on the ground growing, which might be better than no plants. Some limitations of the study. We saw that cold treatment could be acting as some type of stratification, like cold stratification from Helenium grandiflorus, the one species that had higher final germination in the cold treatment. Yeah, we think that maybe that cold treatment, which was 40, I think a low of 4 degrees Celsius, might have been low enough that it was activating that stratification, whereas the other sources might not have been getting that low enough temperature. And we saw an increase in germination due to that. Low germination in some species was likely due to some lack of appropriate pre-treatment, whether that's scarification or other germination methods for breaking dormancy that weren't able to use, and then also low number of sources. This study could greatly benefit from being reproduced with having multiple sources and maybe less repetitions, but including more seeds in those bags. And that would allow one to really look more into the details of local adaptation and those effects of sources. With 3, it's hard to tell exactly what's going on there. So implications for management are that these results can inform decisions for folks in seed. Source matters for some species, but maybe less for others. This could mean that restoration practitioners might want to be more cautious with those affected species and where they get those from. But of the ones that didn't show that interaction affecting local adaptation or presence of local adaptation, this might indicate increased availability of seed that they might be able to use to their advantage for establishment. These warmer conditions induce germination. Could be a type of pretreatment people could implement as it's like slight warm stratification before they add those seeds to the field. And the main point that I kind of want to drive home with management is that there should be an implementation of multiple sources because these can apply to a wider spread of traits. But this is provided that when you're using lots of different sources, there's only 1 or 2 introduction events. As you keep adding more genetic material, you're almost kind of flooding that local adaptation that is present and could lead to some outbreeding depression of that population, that restoration. When using multiple sources, this allows restoration practitioners to plan for changes in climate in multiple climate directions, not just warm. You could go in direction of drier, cooler. As we know, like climate change is becoming just more extreme events, not just warmings. So, and it also greatly lowers the risk of inbreeding depression by having multiple genetic sources. And this is a table that kind of summarizes those effects, or if those species might be affected by transfer, with the ones that have both final germination and germination rate having an interaction. They are definitely likely to be affected by transfer, whereas we find less support for the 3 species that didn't have any interactions of either. They might be okay to transfer, not as affected. And with the sources of 1 of the 2 may be affected, would be worth looking into if you want to use these species for restoration and further research. But that's all I have, and I will open the floor to questions.
Yes, great presentation. I'm just curious about, did you know the variability, the seed variability for all of your seed source from local south and north? Because you have 2 species, no germination. I'm just curious about that. Seed variability.
The variability in the seed
Viability.
viability, excuse me,
yeah,
okay.
Yeah,
Viability, there was viability listed on the packaging of the seed that was commercially bought.
Okay.
But we didn't perform additional TZ tests to look at the viability.
okay.
So
Do you think that could be contributed to
I think it
your
definitely
2
could.
species doesn't have any germination?
I think ideally, some studies have done, like, they do it before, and then if the seeds that didn't germinate, that can be still not broke out of dormancy, then they can open those up and do a TZ on those to see if it's that they're actually viable and that they can remove those from the study, or if they are just dormant. And
Thank you.
yes, Josh.
Can you go to your graph of latitude versus germination?
Oh,
Oh, it's there. That was good with random clicking. So I'm concerned that this analysis is pseudo-replicated.
I think it was you trying to do a like something that shows, but like I couldn't figure out exactly how to make everything made the assumptions, and this one like worked, but when I looked at the independence of variability, that was like high non-independence of those, so
Yeah.
I'm definitely looking into.
So how many data points go into the calculation of the correlation coefficient from which the p-value is derived from? That's
Yeah.
the main
So,
problem here, and I think all of those data points are going into the calculation of the correlation coefficient.
right,
And this is inherently an examination of the variation in germination among your sources, not among the species within your sources or the replication within your species within your sources. So it's not a particularly challenging fix. But I do think it will probably knock the p-value out of
yeah,
significant, because like you said, you don't have that many sources.
yeah, if there was multiple sources, you wouldn't see as many of these lines, like these vertical bits, and is that what you're getting at mostly?
Well, yeah, I mean, the vertical bits simply come from the fact that you have,
Terminology.
that you have multiple species within a single source. And I think is it multiple measures of germination per species, or is it one single measure of germination per species?
It is just the measure of, species were not included as part of this model, so like, it's just, like, every single point is a bag, so,
Okay.
yeah, every single point is a.
Okay. Okay, it's doubly pseudo-replicated.
Yes,
Probably, yeah.
yes.
Yeah.
Cause there should be one average per species, per
Okay.
whatever you're looking
Per,
at.
per.
And then
source,
you
per source.
have to have, you
Yes.
can't just do a correlation, you have to have the species in it's basically like in a regression model.
Yeah.
So you need a more complicated model
Yeah,
to
and that's
handle that.
part of where I'm moving forward, doing, so. I really wanna get into some more modeling when doing this, trying to get this paper out towards publication, obviously, we're gonna have to address some of these, problems before sending that to people. So, and that's modeling both with, not just this question, but also the ANOVAs incorporating more appropriate, Helenium or like.
Yeah, that was gonna
mixed
be,
model.
that was gonna be my other, I don't wanna dominate the conversation, that was gonna be my other suggestion, is that when you go to those rank ANOVAs,
Mhm.
you're basically shooting your power in the head. So if
Yeah.
if you can use an alternative distribution or even a transformation, to get closer to a normal, then you retain a lot more of that statistical power, and some of those differences might pop back out.
Yeah, I think there's definitely a lot of revisions to look, or not a lot, but like some revisions to definitely into future analysis. Yeah. Thank you.
So you didn't show much evidence of local adaptation at all. So this is really interesting to me. I'm just saying, so my take home message is, it looks like it varies, but there's no clear patterns, but that gives you a lot of breadth when you're thinking about management.
Mhm.
Yeah.
Right? So essentially it could, because you were worried about inbreeding or outbreeding and so forth, but the idea is, it looks like you could put things together in these sources, have this genetic variation, and you might have greater probability of long-term persistence.
Right.
I'm just saying, so that's really interesting, because you've done quite a nice breadth of different species.
And it's better for restoration, because they don't have to worry quite as much when the seeds come from.
Exactly, absolutely.
I would be concerned if I wouldn't recommend, that's why I say like recommend a multiple source. I wouldn't recommend saying like, oh, I'm just gonna grab a species from, or a source from wherever, and that's the only one I grab, I would not recommend. I
Well,
would definitely use kind of some redundancy of sources.
So you always want multiple sources, but you want them to be unique. If they're not unique, it doesn't matter,
Mhm.
right? So
Yeah.
this is the other tricky part.
Let's see if
And
there's
all of
anything.
these are very, very widespread species, right?
And most of them are very broadly distributed.
Right, which adds to the
Yep.
power of being able to restore.
Oh my gosh. How do I get the little, the thingy of it?
If you click on the little,
You might have to
Yeah.
drag the mouse all the way to the right side.
Why don't you just do it?
Oh, I'm already here. You know. Yeah. Question, not really. So the question that Brianna has is that, or the question, or the chat that she made. The question is not really about seed germination, but habitat restoration. I've been reading about the land use spillover effect, where restoring natural habitats in one region can benefit the local biodiversity, but it can also have unintended negative consequences elsewhere, particularly where it displaces agriculture, or other land use activities. I'm curious what your overall thoughts are on this.
Hate to restore something that's ag, is that what that's saying?
I guess I'm not familiar with this land use spillover, but I don't, I guess I don't permit or like promote that we should go be like guerrilla like tearing up like places and like crops that like that people use. Obviously that would have legal issues, but
oh, that's wrong.
but I do think there's something to be said for, you know, marginal places that maybe aren't making it much agriculturally that could be maybe better used for restoration and storing some carbon. But does that answer your question? I know that was really vague, but
I guess so. I mean, it was just something that I just read about the other day. So it's essentially saying that, like, you know, an area of agriculture that we're restoring for prairie habitat, we're then losing that agriculture and forcing that agriculture to potentially happen somewhere else in an area where it hasn't happened before. So then you're losing the diversity in that area.
yeah, that's really interesting. I
Yeah, I'd
thought
never
that
heard of it.
before. That's kind of concerned.
Yeah, it's essentially passing the buck potentially.
Yes.
In your discussion, you mentioned some of your species maybe having slightly different results because of like stratification or lack of stratification. So I was just wondering, like, when you were like setting it up, like what kind of went into the decision to like not, or to, you know, like pros and cons of stratifying before this experiment, kind of like
Yeah.
that thought process.
So with getting the seed from commercial providers, we don't know what, if any, like their storage conditions were. And since we're getting those sources across all through temperature treatments, we assume instead of trying to like doubly apply some things that may already be have occurred, that we just keep those species or those sources as they are, and which is what a lot of actual restoration practitioners tend to do, or have been seen to do, where they might just buy the seed and then plant it on the site and hope that it gets germination that year. If not that year, then maybe survives, stratifies over winter. But
Thank you.
So you said that you took all the moldy seeds out of your analysis. So do you mean in all the bags, if any seeds had mold on them, then you remove them from the analysis, or what do you mean?
removed from the analysis in that I reduced that number of seed, since that seed obviously got molded to the point where it was like mushy and not
Yeah.
viable. Removed that seed from like the total numbers of seeds in that bag.
So like if there was 25, then you used 24, and one
Yep.
was moldy, then you did 24. Okay, but like if a seed was moldy, but it also had germinated, that's still viable and you use it in your analysis.
Everyone that I saw that developed a type of like bacterial or fungal infection was definitely very
Not viable.
non-viable.
Not full, as a, okay.
How many seeds were in your beach bag?
25,
25 and then 5 bag. Okay, great.
a total of 125 seeds for combination.
Purse.
Well, 25 times 5, but yeah,
Okay.
it's the number of seeds that were required, because that was part of the restrictions of the species selection, was just finding enough seed to incorporate for the study.
How many seeds went moldy?
It depended on the species. There were definitely certain species and certain sources that would mold more than others. Yeah, I guess the worst ones, I think the worst source species at one point, like all the bags in that temperature treatment had only half of the seeds, partly because I think some of the seeds that were placed in the bag might have been viable in the first place. As far as like being empty seeds, I had a lot of great help putting seeds in bags, but I won't deny that it's definitely hard when you're looking at a seeds on a piece of paper for 5 hours and making sure that you're not putting empty seeds. That was another thing. Empty seeds that I noticed were also removed from the total number.
And you can see that like with grass seeds, like some of them look like
Yeah, it's
pieces
like
of grass
grass
and some
sometimes
have a
a
little
little bit,
hard
'cause
bit
they're
at the bottom.
too hard.
Right.
But if there's like any bit of like softness or give to it, then it's usually not built.
Did you have more mold in the colder temperatures?
No, which is
I
interesting.
was just curious, yeah.
I guess I didn't, there wasn't a lot of times where like, I guess most of the times, yeah, like the filamentous was always the mold, like, but there's also bacterial infections too, but I guess I didn't notice that there was more like filamentous infections versus bacterial like mold, but that would be interesting.
Any more questions?
Any more questions from online?
So then none of these seeds required any formal scarification or anything, nothing.
I think definitely the Dalea was one that could have scarified, but we'd already started that species in hindsight, unfortunately.
Well, but that's not what restoration people would do
Right.
either. So we were just kind of, they just get the seed and put it out,
And
so
they
we just
just-
got the seed and put it in.
No, I was just curious.
Yeah.
And you were basking and basking.
They are, well, the names are, they are BC, Germination and Dormancy, godmothers, if you will.
And godmothers.
Oh, come on, parents.
Godmothers, excuse me, godparents, there we go.
The book is this big now.
It's large,
amazing.
yeah.
Okay, good.
All right, if there's nothing else, we'll go and take a break. Thank you. Don't let go on your way out.
Yeah, I got some food up there.
That was interesting.